Welcome to this interactive study breakdown on biodiversity. Biodiversity is the variety and number of different species in an area. It is a key indicator of ecosystem health. In this video, we will explore how to measure abundance and distribution using quadrats and transects, examine human impacts like pollution and introduced species, and discuss conservation methods. Let's dive in. Let's map out the core concepts. Biodiversity is measured using sampling techniques. Quadrats give you the number of organisms per area, while transects show how distribution changes across a gradient. Human activities threaten biodiversity through habitat loss, fertilizer runoff causing eutrophication, and introducing alien species. We can monitor health using indicator species, and protect areas through conservation. Turning to our core study notes, biodiversity is exactly defined as the variety of species. Abundance and distribution are quantified through sampling. Indicator species like lichens tell us about air quality. Alien species can outcompete natives. Eutrophication is a key example of pollution reducing aquatic biodiversity. Memorize these definitions for recall questions. Now, the process of measuring biodiversity. First, use random coordinates to place a quadrat to avoid bias. Within the quadrat, count individuals or estimate percentage cover. Repeat this many times to get a representative sample. Finally, calculate the mean abundance. For distribution, use a transect line with quadrats at regular intervals. This method shows how species change across environmental gradients. Let's work through a typical exam question. Fertilizer runoff adds nutrients, causing an algal bloom that blocks sunlight. Submerged plants cannot photosynthesize and die. Bacteria decompose the dead plants, using up oxygen. This creates dead zones where fish and invertebrates cannot survive, drastically reducing biodiversity. Remember to state each step clearly. Watch out for these common pitfalls. Many students mix up abundance and distribution. Abundance is how many, distribution is where. Always state that quadrats must be placed randomly. When using indicator species, specify which pollutant they indicate. For eutrophication, the key is oxygen depletion causing death of aquatic life. Examiner advice: use precise terms and mention mechanisms. To wrap up, biodiversity is the variety of life. Measure it with quadrats for abundance and transects for distribution. Human activities threaten it through eutrophication, alien species, and habitat loss. Protect biodiversity by using indicator species and conservation. In exams, define terms precisely and explain processes step by step. You've got this.